Somewhere across the Americas, in the blood of millions of indigenous people, sits a genetic pattern that has no equal anywhere else on Earth. Not a rare exception, not a regional quirk. A near total dominance of a single blood type, type O, found in some communities at rates of 90%, even 100%. Nowhere else on the planet does one blood type claim a continent this completely. For decades, scientists noted it, measured it, and filed it away as an unsolved curiosity. They had theories. They had guesses. What they didn't have was proof. That has changed. Advances in ancient DNA analysis have finally allowed researchers to trace this bloodline back to its true origin. And the answer is not what most people expect. It has nothing to do with coincidence. It has nothing to do with isolated legend. It traces back to a single brutal chapter in human history, a journey across a frozen land bridge, a population cut off from the rest of the world for thousands of years, and a genetic bottleneck so severe it reshaped an entire hemisphere's blood. This is where type O blood in the Americas actually comes from, and why it never should have become this common. Blood may look uniform to the naked eye, but underneath, it is divided by a system most people never think twice about. The ABO blood group. 4 categories: A, B, AB, and O. The difference comes down to antigens, tiny proteins and sugars sitting on the surface of red blood cells, markers your immune system uses to distinguish self from invader. Type A carries A antigens. Type B carries B antigens. Type AB carries both. Type O carries none. It's not an addition, it's an absence, a clean genetic gap where 2 other markers should be. That absence is exactly what makes type O the universal donor. The blood type emergency rooms depend on when there's no time to test a match. Globally, it sits at roughly 62%, the most common blood type on Earth. Common, but not overwhelming. Everywhere else, A and B blood carve out substantial territory alongside it, except in the Americas. Among indigenous populations, type O doesn't just lead, it dominates. Rates climb to 90%, sometimes higher. In some communities, researchers have documented figures approaching 100%, a number that shouldn't exist anywhere in a genetically diverse human population. No other continent shows anything close to this level of concentration. Not Europe, not Asia, not Africa, where blood type diversity runs deepest of all. For decades, this was noted and set aside, an interesting footnote in population genetics textbooks. But a statistic this extreme doesn't happen by accident. Genetic uniformity on this scale points to something specific. A population that passed through a bottleneck, a moment in history so narrow that only a fraction of the original genetic diversity survived to the other side. Something filtered this bloodline down to almost nothing but type O. And whatever that filter was, it happened before written history, before agriculture, before anything we would recognize as civilization. To find it, researchers had to look back to the last ice age, and to a migration that changed everything. When geneticists began mapping blood type distribution across the globe, they expected the numbers to roughly track known migration patterns. What they found instead was a map with one glaring outlier, and it sat squarely in the Western Hemisphere. Start in Africa, the continent where human life began. Blood type diversity here runs deep. A, B, AB, and O, all present in meaningful numbers. A genetic record of the longest, most complex human history on Earth. As early populations pushed north into Europe and east into Asia, the picture shifted. Type A rose sharply across Western Europe. Type B became dominant across Central and Eastern Asia. Type O remained present everywhere, but its share steadily declined the further populations traveled from their origin point, diluted generation after generation by new mutations, new mixing, new pressures. Then the map crosses into the Americas, and the pattern collapses entirely. Here, type A nearly disappears. Type B is almost non-existent. Type O stands alone, claiming close to the entire population in region after region, from the Arctic reaches of Canada to the rainforests of the Amazon. This isn't a gradual decline mirrored elsewhere. It's a wall, a hard genetic boundary that separates the Americas from every other landmass on Earth. Geneticists studying this anomaly kept arriving at the same conclusion. This pattern doesn't reflect gradual global spread. It reflects isolation, a population separated from the rest of humanity so completely and for so long that whatever genetic diversity it once carried simply never had the chance to be replenished. That raises an obvious question. How does an entire hemisphere end up with almost no genetic contact with the rest of the world for thousands of years? The standard model points to one narrow, frozen answer: a land bridge, a crossing, and a journey that sealed an entire population's blood into place for millennia. During the last ice age, sea levels dropped low enough to expose a strip of land connecting Siberia to Alaska, Beringia. For a narrow window in geological time, it functioned as a bridge between two hemispheres that had never been connected before. Small groups of hunter-gatherers crossed it, following game, following the edges of retreating ice, carrying with them whatever genetic diversity existed in their small founding population. That detail matters more than it sounds. A small founding population doesn't carry the full genetic range of the larger population it came from. It carries a fraction, a sample. If that sample happened to be dominated by type O blood, and evidence increasingly suggests it was, everything descending from it would inherit that same imbalance. Compounding that effect further, once the ice retreated and sea levels rose, Beringia disappeared beneath the ocean. The bridge closed. The Americas were cut off from Eurasia almost completely, not for centuries, but for thousands of years. No new populations arriving, no new genetic material entering the pool, just a small founding group, isolated on two continents, breeding within an increasingly narrow genetic range, generation after generation. What started as a chance imbalance in a handful of migrants hardened, over millennia, into a hemisphere-wide pattern. The data backs this up starkly. An influential study conducted in the 1980s examined Native American populations in Arizona and found that 99% of participants carried type O blood. Not a majority. Not a strong lean, effectively total saturation. Similar patterns have surfaced from the Arctic communities of northern Canada down through the dense forests of the Amazon basin, a continuity that spans nearly the entire length of two continents. This is what a genetic bottleneck looks like when you can still see it thousands of years later, written directly into the bloodstreams of tens of millions of people. But isolation and small founding numbers only explain how the pattern could survive. They don't explain why it would have been favored so overwhelmingly, why nature seemingly reinforced this outcome rather than diluting it. For that answer, researchers had to look at what these early populations were actually up against: disease, altitude, and an unforgiving new world. Isolation explains how type O blood survived unchallenged. It doesn't explain why it thrived. For that, researchers turned to the two forces that shaped survival most brutally in the ancient Americas: disease and altitude. Before I show you the last part, if you've ever looked at your own results and wondered where indigenous ancestry fits into your bloodline, I put it all together in a guide called Where Do You Really Come From? Link is in the description. But stay right here, because what comes next is the part most people completely miss. Start with disease. Certain pathogens target the very antigens that distinguish blood types from one another, latching onto the A or B markers on red blood cells to gain entry. Type O, lacking both, offers those pathogens nothing to grab onto. Research elsewhere in the world has linked type O blood to measurably lower rates of severe malaria, a connection tied to the same antigen targeting mechanism. Malaria wasn't a defining pressure in the pre-Columbian Americas, but the underlying principle likely extended further. A subtle resistance against a broader range of pathogens that type A and B carriers had no defense against. In small, isolated founding populations, an edge like that doesn't stay small. It compounds. Then there's altitude. For thousands of years, human populations lived and thrived in the Andes, at elevations where oxygen runs dangerously thin. Their bodies adapted in dramatic ways, and one theory ties blood type directly into that adaptation. Type O blood tends to run less viscous than A, B, or AB, meaning it can move more freely through narrow capillaries. In low oxygen environments, smoother blood flow translates directly into more efficient oxygen delivery to starving tissue, a genuine survival advantage at elevations where every breath counts. Neither theory alone explains the totality of what happened. But, stacked together, disease resistance and oxygen efficiency, in populations already narrowed by isolation, the outcome stops looking like a coincidence. It starts looking like exactly what evolution does best: take a chance imbalance and turn it into an advantage too powerful to reverse. Survival in the ancient Americas wasn't only a matter of biology working in isolation, it was also a matter of diet and of the bonds holding small communities together. Indigenous diets varied enormously across the continent. Seafood-heavy traditions along the Pacific Northwest, corn-based agriculture through Mesoamerica, protein-rich hunting economies across the plains and the Arctic. Some researchers have proposed that blood type may influence how efficiently the body metabolizes different nutrients, with type O linked in certain studies to more efficient processing of high-protein, high-fat diets. The exact profile common among hunter-gatherer societies before agriculture reshaped how humans ate. If that connection holds, it means type O wasn't just neutral against the environment these populations faced. It was suited to it, tuned to the exact nutritional pressures a hunting and gathering lifestyle demanded. Then there's the social dimension, easy to overlook but potentially just as significant. In small, tight-knit communities, survival wasn't an individual project, it was collective. Childbirth complications, injuries from hunting, wounds from conflict. These moments demanded immediate blood transfusions long before anyone understood the science behind why some transfusions worked and others didn't. A universal donor, someone whose blood could be given safely to almost anyone in the group, would have held a quiet but critical kind of value, not measured in status or wealth, but in something more fundamental: the ability to save a life when nothing else could. That value doesn't disappear with time. It compounds across generations, the same way genetic advantages do. A trait that helps a small population survive childbirth, survive injury, survive the ordinary emergencies of a hard existence spreads further and faster than most people realize. Diet, disease resistance, altitude adaptation, and social value inside close survival networks. No single force explains why type O became this dominant across two continents. Together, they explain almost everything, and they set the stage for a new kind of evidence, one that doesn't come from theory or migration models, but from DNA itself, tracing individual bloodlines directly back to the populations who lived through all of it. For most of history, the theories about type O blood's origins in the Americas remained exactly that: theories built on population statistics and educated guesswork. Modern DNA technology changed that. Genetic testing now allows researchers to trace ancestry with a level of precision that simply didn't exist a generation ago, confirming lineages, mapping migrations, and in some cases, rewriting assumptions that had stood for decades. One case in particular drew significant scientific attention. A man named Crawford, living on the Blackfeet Indian Reservation in Montana, a reservation spanning over 1 million acres and home to one of the largest Native American tribes in the United States, underwent DNA analysis that researchers described as revealing an unusually deep and well-preserved genetic lineage. His results offered a level of ancestral clarity that prompted scientists to reconsider parts of the standard migration narrative, the story of how, and even when, early populations first arrived in North America. Cases like this carry weight far beyond one individual. They connect personal identity to a much larger scientific record, tying a living person's bloodline directly to a migration that happened more than 10,000 years ago. For indigenous communities, this kind of confirmation can matter enormously, reinforcing oral histories and cultural continuity that outside institutions had long questioned or dismissed. But the science doesn't arrive without complications. Genetic ancestry testing raises real ethical questions. Questions about consent, about privacy, and about who controls and profits from indigenous genetic information. Being native isn't defined by DNA alone. It's shaped by community, culture, language, and lived history, dimensions no test can fully capture. Researchers working in this space increasingly acknowledge that responsible science here means more than good lab work. It means respecting the people whose ancestry is being studied. Even so, the DNA evidence keeps accumulating, and it keeps pointing in the same direction. A small founding population, isolated for millennia, whose blood tells a story far older and far more specific than anyone could have proven before. And that same technology has surfaced connections even stranger than the land bridge itself. The deepest layer of this mystery doesn't sit in the Americas at all. It sits in Siberia, at a lake older than the story of modern humans itself. Lake Baikal, the world's oldest and deepest freshwater body, has functioned as a genetic crossroads for tens of thousands of years, a meeting point where populations moving east and west intermingled long before anyone crossed into the Americas. Some researchers now propose that populations connected to this region may represent the deep ancestral source for the founding groups that eventually crossed Beringia. If that holds, the story of Type O blood in the Americas doesn't begin at the land bridge. It begins thousands of miles further west, at the edge of a Siberian lake that has quietly shaped human genetics for over 20,000 years. Then there's a discovery that complicates the isolation story even further. Genetic evidence suggests contact between Polynesian populations and indigenous Americans roughly 800 years ago, long before European contact reshaped the continent. Shared genetic segments have turned up across multiple Polynesian islands, evidence, researchers argue, of a real historical encounter rather than coincidence. The presence of American crops like sweet potatoes in Polynesian archaeological sites, absent everywhere else outside the pre-Columbian Americas, only strengthens the case. Somewhere in that era, two populations separated by thousands of miles of open ocean briefly connected, and it left a trace in the DNA of both. And then there's the story that predates all of it, the kind that outlives data. Among some indigenous communities, oral tradition tells of a stranger, pale-skinned, arriving alone from beyond tribal lands, his presence tangled ever since with the appearance of Type O blood in certain family lines. No record confirms who he was, or if he existed at all, but the timing, in the stories at least, lines up in a way that has never fully faded from memory. Science can trace a bottleneck, it can date a migration. What it can't always do is explain why a story like that persists, generation after generation, refusing to disappear